Okay, let's grab our magnifying glasses because today we are putting on our detective hats to investigate a truly foundational framework. We're doing an explainer on the source text for the polymerase chain reaction protocol. And look, we aren't just geeking out over the theoretical chemistry today. No, we are actively decoding how this vital text sets deliberate, totally non-negotiable boundaries for anyone even attempting this technique. Actually, when you really look at the architecture of this document, it reads almost like a strict legal contract for molecular biology. It establishes a crystal clear path, setting the exact parameters you absolutely need to successfully replicate DNA segments. Now, for those of us who already follow this field, we know how notoriously sensitive these reactions are, right? But what's really fascinating about analyzing the source material itself is seeing how the authors literally built that sensitivity straight into the formatting and the language. So we're going to explore exactly why the document is structured the way it is, and how grasping that structure really unlocks the precise mindset you need to operate in such a high-stakes biological environment.
Okay, let's dive into this. Our source material immediately isolates the core subject, the polymerase chain reaction. But here's the catch. It frames it strictly as a protocol. It totally bypasses the theoretical chemistry right out of the gate to establish a really firm baseline. We are dealing with a highly structured, uncompromising procedure here. Now, in the laboratory, the word protocol carries immense weight, doesn't it? It implies that the variables have already been tested, the margins of error have been calculated, and the optimal path has been definitively mapped out for us. So by labeling PCR this way, the text is practically shouting that success isn't about improvisation. It's not about creative problem solving at the bench. It is entirely about adherence. If you understand PCR just as a biological concept, well, you really only know half the story. The document insists you've got to approach it as a rigid set of operational rules. It's a specific technique where precision isn't just encouraged, it is the absolute prerequisite for getting any valid result.
Now, what's really interesting about this slide is how the exact phrasing in the source takes this a whole step further. It explicitly defines the procedure as a PCR technique step by step. Notice it's not presented as some loose set of guidelines or, best practices you can just tweak on the fly. By hard-coding that phrase, step by step, into the framework, the text is telling us that the order of operations is literally the defining characteristic of the entire process. This kind of sequential dependency, it means that step 3 absolutely cannot succeed if step 2 is even slightly off. Every single phase relies entirely on the precise execution of the phase right before it. It essentially creates a single point of failure at every single juncture. As an analytical framework, this tells us that when you're troubleshooting a failed reaction, you're not usually rethinking the fundamental science. Instead, you're retracing your steps, acting like that detective again, to find the exact moment the sequence was compromised. The text completely elevates the concept of a strict sequence from a mere suggestion to a hard operational mandate.
So the crucial point is how this framework sequentially narrows our focus. It establishes the big overarching goal first, the polymerase chain reaction protocol. This is your theoretical objective, right? The successful amplification of a specific DNA segment. But then, immediately beneath that, the document dictates the physical reality, PCR technique step by step. The source text demands that to truly grasp the big picture protocol, you just have to completely surrender to the sequential execution. It's this fascinating duality in the text. The protocol represents the macro-level understanding of what we're actually trying to achieve, while the technique step by step represents the micro-level discipline required to actually get it done. You literally cannot have one without the other. The document cascades its information this way to carefully condition your mindset. It forces us to acknowledge the grand scientific goal, but then instantly grounds us in the cold mechanical reality that getting there requires executing a sequence of highly controlled, very specific physical actions.
As we wrap up this explainer, I want you to imagine something. Imagine you're a new trainee stepping up to the lab bench for the very first time. Your pipettes are calibrated, the thermal cycler's good to go, and your samples are all prepped. But before you ever even touch a piece of equipment, you have to answer this fundamental question: why is a strict sequence so crucial for PCR success? Well, our source emphasizes this precise framework because in any strict protocol, improvising even a single step fundamentally destroys the outcome. You aren't just following rules to be neat and tidy. You are manipulating molecular biology on a microscopic scale, where even a tiny microscopic deviation in temperature or timing is going to yield a completely false narrative in your data. So, I'll leave you to ponder the profound importance of that structured methodology, because when the stakes are this high, your greatest tool isn't just your scientific knowledge, it's your absolute discipline to the sequence. Thanks so much for exploring this framework with me today.